Thank you very much, Melina, for the kind introduction. I will first try to share my screen. Can you see it? No?
Yes, it's coming. Yes, we can see the screen.
Yeah.
Thank you.
Okay, perfect. Good afternoon, everyone. First of all, I would like to thank all the organizers for inviting me, but also for preparing such an amazing event, because so far we have seen really, really good presentations. So my second thanks goes to all of the participants. And I'm excited to hear even more for the rest of the conference. And for today's session, I have prepared a presentation about environmental monitoring with focus on assessing the bioavailable trace elements in impacted environments. But first, I will start with a simplified overview showing evolution in environmental pollution and related monitoring. Naturally, a big boom in environmental pollution is related to industrial revolution in the early 1900s, which is related to enhanced release of inorganic pollutants. Now, this is further followed in the 1950s by the increased use of pesticides such as DDT and PCBs.
Sorry, Dr. Smolíková, sorry
Yeah.
to interrupt, but we see a gray area on top of the presentation.
Sorry?
We see a gray area on top of the presentation. I don't know if there is something that it's moving.
Oh, I don't see anything.
I would say it's the zoom bar when you are showing with zoom, it covers the titles, okay?
I see. Yeah, okay. I will just not use the pointer then. Is it still there or
maybe you
you
can
might
share
try to share the not the desktop but the screen of the presentation.
I am actually sharing the screen, not the desktop.
But okay, right now it's a very small gray area, so
okay, because
we can
I
continue.
don't see that at all. But I can still try to change it to share the other. Is it still there?
It's still there, but it's very, very small now. So please continue.
I will just not use the laser pointer because I think if I cross the screen, then it's appearing there. Okay.
Thank you.
Yeah, sorry about this. So yeah, as I mentioned, in the 1950s, there were first concerns arising regarding the radioactive waste. Then further in the 1970s and 80s, the focus shifted more towards the air pollution, air quality related to the presence of acid rains and also the release of greenhouse gases, which caused the depletion of the ozone layer. And of course, since the early 2000s, we have this biggest issue that everybody is worried about, which is the climate change. But also from the monitoring point of view, we have a lot of emerging contaminants such as microplastics, nanomaterials, or pharmaceuticals, and so on. Now, this increasing trend in environmental pollution is naturally related to intensified efforts to monitor the quality of our environment. And as you can see on this nice illustration from Beck et al., who used the data from the European Environmental Agency, the water quality monitoring evolves geographically, but as well as over time. Of course, we can see very clear increasing density of monitoring activities over the displayed 40 years. But we can also notice clear expansion to Eastern Europe after the end of the Cold War. However, regardless the changing trends, the trace elements still remain among the significant global challenge due to their toxicity, persistence, and bioaccumulation potential, which is why I'll focus on them in today's presentation. While elevated concentrations of potentially toxic elements are generally related to anthropogenic activities such as mining, industry, or agriculture, we can still find natural hotspots which are related to unique geological backgrounds, because trace elements are naturally occurring in the environment, and this is very important to keep in mind. And I will come to that a little bit later in my presentation. We, of course, also need to distinguish between essential and non-essential trace elements. The essential ones are a little bit tricky because there is actually very thin boundary between their essentiality but also toxicity, which is generally the matter of their concentration. For this reason, there is a lot of environmental monitoring programs adopted on national as well as international level. With regard to water quality monitoring, these programs generally follow the European guidelines set by Water Framework Directive. However, this conventional monitoring is generally performed by discrete sampling. Now, what does it mean? You're all familiar with this. Imagine you want to sample water in this nice river. So you would just go there, take your water sample, and ideally directly on the side, you would filter the water on a pore size of 0.45 micrometers, and you would transfer your sample back to the laboratory for analysis. But I want you to imagine that the concentrations in your river water are fluctuating in time. This can be due to the presence of discharge from industry or due to tidal influence in estuaries or coastal zones. Now, depending on the time of your sampling, you would get very different results, which are actually misleading and not representative for the environment that you are studying. So to avoid this, you would have to perform sampling in time series. Now, you can either do this by spending a really nice day by the river and sampling the river water every hour for over a period of, for example, 12 hours. Or in certain cases, such as, for example, monitoring the quality of effluents from industries, you can apply these automatic samplers, which are collecting the water at preset periods. And you can obtain these several spot samples. From these, you can then calculate the time-weighted average of discrete sampling. However, you can notice on the right side that between these two spots, you still miss a high peak, and therefore you are not fully capturing the representativeness of the pollution load in the area. And therefore, there is an increasing effort to use alternative approaches, which are passive sampling methods. There are actually many of them, but I will only mention today the one that is specific for trace elements and which is also my specialty, and that's the diffusive gradients in thin film technique, or briefly DGT. So basically, to use it, instead of just taking your water sample, you would deploy your passive samplers in the river water and leave them there for a certain period of time. This is typically 24 to 48 hours, but it can be even up to a week or two, depending on specific cases. And you would just let the samplers to accumulate your analytes for you during this whole time. Then you take them back to the laboratory, extract your analyte from the samplers, and you will calculate this DGT determined concentration, which is full representativeness of your pollution in the area. Beside that, the DGT technique has other advantages, such as pre-concentration of the analytes directly in situ, which means you can analyze higher concentrations that normally your analytical techniques would not be able to capture. You're also conserving the analytes directly on the site, so there are no changes during the transport or storage of your samples. You're also eliminating the metrics of the sample, which can be really beneficial, for example, when you are analyzing seawater, which is very difficult to measure. And you are also getting information about the bioavailability of trace elements. Now I would like to briefly talk about the DGT technique itself, because it is not very common and people usually tend to not really understand it when they're only reading about it. It is not that difficult, but there are some specifics about it. So as you can see on the left side, that is a DGT sampler, which consists of plastic housing, which incorporates 3 specific layers. And that's a protective membrane filter and 2 types of hydrogels. The diffusive gel, which serves as a medium for transport of your analytes from the bulk solution, towards your resin gel, which has some sorbent incorporated in its structure with functional groups that have high affinity for your analyte. Now, when you have all those 3 layers, you will just enclose the unit, which has exposure window on the top, and you will deploy the samplers in your water. This doesn't have to be just water. These piston shape-like units can be used in water, but also soils. And the long probes that you can see on the left are actually meant for studying the depth profile in sediments. Basically, you insert them in the sediments, and after retrieving them, you cut the gels in slices so that you can see the depth evolution of your trace elements. And you can better describe their cycling between water and sediments in the area. And now for the principle of the method. I'm pretty sure every one of you ever heard about diffusion. So it's a movement of molecules or ions, generally solutes, from areas with high concentration, which in this case are represented by the bulk solution. And the analytes will move to the areas with low concentration, which in this case is represented by the diffusive gel. Because the gel itself consists from 99% water, so it kind of represents a solution as well. And now the diffusion would normally continue until there is equilibrium reached on both sides, on the diffusive gel and bulk solution. However, the trick of the DGT technique lies in the presence of this resin gel, which, as I said, has some sorbent with functional groups, which are progressively binding and removing the analytes from the solution, which creates a flux. Meaning that the concentration from the solution is gradually decreasing and reaching 0 on the interface of these 2 gels. And because we are creating flux, we can quantify it using a first Fick's law of diffusion, which is modified here for calculation of the DGT determined concentration. And now I'm getting to the bioavailability, which I have already mentioned a couple of times during the presentation, but I would like to explain what does it mean in the sense of this presentation. So first we need to have a look at the distribution of trace elements in the solution, which, as I mentioned before, according to the Water Framework Directive, you can distinguish between particulate and dissolved fraction of trace elements by filtering the water on 0.45 micrometer size filters. And then we are only quantifying the dissolved fraction. However, if we look a little bit further, we can still distinguish between 2 phases within the dissolved fraction, and that's truly dissolved and colloidal fractions. And the truly dissolved fraction is represented by weak complexes and free metal ions, which are generally considered to be able to cross the biological membranes and therefore are potentially bioavailable. In other words, if you are analyzing water from areas where you have a presence of non-labile complexes, these may decrease the bioavailable fraction of the analyte, and therefore whatever you are analyzing at its dissolved concentration does not necessarily mean it is available for living organisms. Now, there are different approaches which can be used to separate this fraction. For water samples, the most known one is the ultrafiltration, which is a series of different filters, different pore sizes. And the general consensus is that the labile fraction is whatever passes through a filter with 10 kilodalton size. For the solid samples, you can use sequential extractions. These are usually a modification of the BCR protocol specific for different analytes that you want to separate using different solvents. Or, of course, you can perform direct experiment by looking at the uptake of the trace elements from the environment by living organisms. These can be plants, animals, anything. Or what you can do is more elegant solution to use the DGT technique, because this technique actually directly discriminates for the mentioned truly dissolved fraction, and therefore anything that you measure by this method will be potentially bioavailable. There is a general effort among researchers to adopt uniform approaches that will be able to determine the bioavailability of trace elements. The question is the DGT technique able to be implemented in regulatory guidelines? to answer this, I like to use this study from Rougier, which performed a complex evaluation of the suitability of DGT technique for environmental monitoring. So basically in their study, they are sampling at 17 sites in southwestern France, and they do this in the course of 6 campaigns. And they are comparing the DGT technique with the conventional sampling, so just grab water sample. They focus on a wide range of analytes, divalent, trivalent metals, but also oxyanions such as arsenic and selenium. And there are certain outcomes of this study which I find interesting. And first of all, it's some of the drawbacks that are mentioned in this study, and that's you always have to keep in mind that there is a possibility of losing some of your samplers. They accounted 3% loss. This is because if you deploy DGTs in areas where there is free access to people, if they usually see something in the water, they will take it out. So you need to be very careful when you work with this technique. And another thing is repeatability, which was found in this study to be less than 25% for majority of the trace elements. However, for some elements, this is a little bit higher, 41 to 65%. But in my opinion, this is not really good comparison, because in the study, they only use duplicate of the samplers, which is against the traditional guidelines for the use of DGT technique, which asks to use at least 4 replicates, because you might get a damage of the sampler, because if you deploy them for 24 hours, there is always a chance that you will lose it, or it will be damaged, it will be covered by sand or dirt. So it's always better to use a higher number of replicates. And what is novel and very interesting is that this study includes the economical aspect, the cost comparison between the use of DGT with conventional sampling. And they included the material costs, field operations, analysis, and data processing. And they concluded that the DGT application costs overall 2 to 3 times more as compared to the normal conventional sampling. However, in this study, they took into account the costs of the DGTs that are produced by the DGT research company, which you can purchase already pre-assembled. But this cost can be actually significantly reduced if you are manufacturing the DGTs yourself, which is possible. And I would like to stress that even though the cost is higher, you need to realize that, first of all, you are getting additional information about the bioavailability. And second of all, you are getting the time-weighted average concentration, as I mentioned at the beginning, because in this study, the DGT results were compared with 2 grab samples taken at the beginning and at the end of the DGT deployment. And now I would like to demonstrate the application of the DGT technique in the mining context on our case studies. Now, these studies are performed in collaboration with Orano, which is a French corporation covering the whole nuclear cycle from mining to production of nuclear fuels. And our overall aim in these studies is to perform geochemical sampling of surface waters in regions with historical mining activities. We focus on the monitoring of uranium concentrations, and we look at them in relation to the natural background and bioavailability in order to be able to distinguish the environmental footprint of the mining activities from the geochemical baseline. And this is because, as I mentioned at the beginning, inorganic elements are naturally occurring, and the regions of mining sites will likely have higher concentrations of the uranium that we are focusing on now as a reflection of the local geology and hydrogeochemistry. And for this reason, we focus on a thorough selection of the sampling sites. So in collaboration with hydrogeologists, we identify sampling sites located upstream and downstream from the mining sites. But thanks to the collaboration with Orano, we also have access to the mining sites itself so that we can directly sample the mining waters. And on top of that, which is very important, we are sampling also the unimpacted natural watersheds to be able to define the natural background concentrations, which is often not taken into account in this kind of studies. Now I will briefly show the strategy of the campaigns. So of course we focus on the sampling. We perform the water sampling according to the Water Framework Directive, as in filtered by 0.45 micrometer filters. And we split the sample for determination of trace elements, major cations, anions, nutrients, inorganic, organic carbon, alkalinity. We of course measure the pH, temperature, redox, conductivity, everything. And this is my recommendation to everyone. If you are doing any environmental study, take as many data as you can, because it will help you to explain and interpret your obtained results. Then, of course, we focus on the DGT deployment. In these studies, we use specific DGTs for the determination of uranium, radium, divalent metals, and at selected stations, we also deploy DGTs for oxyanions. And all of these DGTs are deployed at 4 replicates for each site. And why do we collect the samples and analyze everything? Well, this is because we then use the results and we use them for geochemical modeling using FreeQC with the ProData database, which is thermodynamic database dedicated to uranium mining activities. And using the data from modeling, we are able to define the speciation of uranium in the studied waters, which can in turn help us to better understand the obtained DGT results. And by using this combined approach, we're able to completely evaluate the trace elements geochemistry in the mining context. Now here you can see the areas of interest. We have performed 4 big studies so far in France in the former mining areas between 2020 and 2024. And as you can see, we have always selected between 44 to 84 stations. So these studies are much bigger than the one I mentioned, just evaluating the applicability of DGTs for these kind of studies. And because we deployed 4 types of DGTs with 4 replicates, we are getting up to a number of about 1,000 DGTs per campaign. And these campaigns were actually performed over the course of 2 weeks, usually. So in one go, we didn't go back, we just did everything at the same time. And here you can see the comparison of the DGT labile and dissolved concentrations of uranium in the Loire region. So it's from the first campaign in 2020. And here is the comparison of all 4 types of waters. So on the first side, you can clearly notice that there is basically no difference between the waters located upstream from the mining site and the natural watersheds, where the concentrations are generally below 6 micrograms per liter. As expected, the highest concentrations were then found at the mining waters, because these are basically sampled directly at the former open pits or discharges of treatment stations. So we expected to see the highest concentrations there. It is important to mention that this graph has a logarithmic axis and the scale is logarithmic. So although the DGT results are seemingly comparable with the dissolved concentrations, it is not really the case, but I will get to it on the next slide. And if we look further on the downstream waters, we can see a very rapid decrease in both uranium fractions, which is especially evident for the site Chardon, which had the highest concentration on the mining side, but further downstream, the concentrations are comparable to the other sites. If you look more into detail on the bioavailability of trace elements beside uranium in the Loire region, we can see that some elements are predominantly present in the colloidal form, as highlighted in yellow in the table, while other elements, including radium and uranium, are rather present in the labile form. Those are shown in blue. However, it is important to mention that the smallest labile fraction of these 2 elements were found at the mining site. The presence of colloids in the waters is strongly influencing the lability of trace elements, as confirmed by the results of saturation indexes, which were performed using the FreeQC software, which pointed out that the importance of iron and aluminum hydroxides on the control of uranium in the mining waters. And when we look at our results at the regional scale, we found that the elevated concentrations of uranium do not necessarily correspond to those of radium, However, the highest concentrations of both elements were found naturally at the mining sites. The overall impact of the mining waters on the environment is, however, minor, because the concentrations of uranium and other trace elements further downstream are below 3 micrograms per liter, and so well within the legislative standards. If we look at the previously reported journal relationship between crystalline basement and the bioavailability of uranium in European waters, we did not observe this same trend in our study. And this is because although the Clisson massif where the sampling was performed has crystalline nature, the presence of carbonates in the waters controls the uranium speciation mainly by carbonate complexes, which results in overall lower bioavailability of uranium than expected. And just to summarize the whole presentation, I need to stress that the regulatory standards fail to consider the bioavailable fraction of pollutants, which is, in my opinion, essential for accurate environmental assessments. This results in the need to use alternative approaches such as passive sampling, ultrafiltration, or sequential extraction in order to get a deeper insight in the lability of trace elements. And finally, as shown on our case study, the combination of alternative sampling approaches and geochemical modeling contributes to deciphering the relationship between natural background and the footprint of mining activities. The gained knowledge can be further used to improve the mitigation strategies and remediation of anthropogenically impacted environments. Now, in the end, I would like to thank to all of my collaborators here from VUB, but also from France, Orano Mining, and Cabinet Relais. And I would also like to thank you for your attention, and please feel free to ask me any questions.
Thank you very much, Vendula, for this very great presentation, very interesting. So I think that we have seen some questions in the chat, our comments. The first comment from Ariane was that maybe Alekos wants to comment on the 1980s Chernobyl event that gave the idea for the Forex Atlas. So maybe it's a question addressed to Alekos. I don't know if he wants to answer. Or maybe not. Okay, so maybe we move to the next comment.
I'm trying to.
Okay.
I'm sorry. I'm trying to copy a message, but I cannot do it. I don't know why.
Oh, on the chat, you mean?
Yep. I'll send it to the webmaster.
Okay. Okay, thank you. So Ariane is also saying that she thinks that in slide 6, Vendula was referring to bioaccessibility, potentially because you said potentially bioavailable.
Yeah, okay. Yeah, my mistake. Yeah.
Okay. Then Nick is saying in ferrihydrite, we don't actually know the chemical structure. For example, I don't know if this is addressed to you.
Yeah, I'm not
Is
sure.
It? Not sure.
If it is, maybe it was the saturation indexes, but these were done by the modeling of the composition of the waters. And this was the composition that we got out. It wasn't ferrihydrite, it was iron hydroxide that I mentioned directly.
Thank you. Carlos is saying, thank you for your excellent presentation. Kelex 100 has a limit of 0.5 milligram per liter for the concentration of elements, which is easily attained in highly contaminated areas, such as mining areas. How does the other resins mentioned behave? High concentrations of other elements may interfere with your target elements?
So I think this is targeted to the uranium. Basically, in this study, we are using specific sorbent for uranium. It's called Defonex. It has been discontinued, but it's been replaced by Monophos. It's from Triscam company. And this sorbent has incredibly high sorption capacity for uranium, and it's not influenced by basically anything. It doesn't sorb many other trace elements. It is working over very wide pH range, even acidic waters. So it is actually ideal to be used in the mining sites. And that's why we use 4 different sorbents. So we have one for uranium, one for radium, and then Kelex used only for the other trace metals, which we are using to see the bioavailability of the other trace elements.
Okay, thank you. So Alekos has written a question. Have you consulted the FOREGS, Geochemical Atlas of Europe? The atlas gives the geochemical baseline of Europe for stream water, stream sediments, etc. It covers 26 European countries, including France, and uses a harmonized method of sampling, sample preparation, and analysis. Also, please be careful with the use of the term pollution, which is used when there is sound evidence that there is a harm to the environment and organisms, including humans. So have you checked this?
Yeah, absolutely.
This atlas?
These studies are actually a follow-up on the FOREGS work, of course. And as I've shown even in the end, comparing the relationship between crystalline basement and the bioavailability of uranium, that's also based on a study that was done on FOREGS data. So yeah, we are of course aware of this, and we are using the data from this database as well.
Okay, thank you. From another question from Ariadne, does DGT works also with seawater?
Yes, it does. Basically, the DGT can be applied in various matrices because then you are, of course, eliminating it for your analysis. Then it depends on your sorbent which you are using. So for uranium, it works perfectly because none of the elements that are in the water will be analyzed, will be accumulated, and won't be interfering. For Kelex, we do some studies on seawater, on a Belgian estuary here, on coastal waters. The only issue is because Kelex, which is used for divalent metals, is basically binding almost everything. You have to be careful about reaching the sorption capacity. So the deployment times are usually around 24 to 48 hours. But with the sorbents for uranium, we have also done very long deployments in seawaters for up to 28 days.
Okay. And by curiosity, what's happening if you're letting this DGT for more time?
Well, there might be issues occurring in some waters when you start to have biofilm forming in stagnant waters, for example. So we need to be careful about that and take it into consideration. But this is usually in stagnant waters, ponds, lakes, where you also have to take into account that there is no flow and take it into consideration for your calculations.
Okay. And I was also wondering if you have already compared the result that you can obtain with the DGT passive samplers with other techniques that may measure the potentially bioavailable fractions, such as voltammetric techniques?
In this study, we did not, but there is another very, very big project. It's called Monitool, and it was done by many universities and researchers along Europe, and it was study on coastal areas where they compared the DGTs with voltammetry and conventional sampling by crab waters samples, and this study was comparing it. We did not do it in our study.
Okay. Carlos, maybe you want to add something
No,
about
no,
Monitool?
well,
No?
because we are on time for the break. Yet it's true what Vendula is saying. Monitool did a lot of work comparing these with another analytical techniques, which is really important to study the bio